This conference will now be recorded.
Hello.
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 Yes, doctor. Huh, doctor.
Is it audible now?
Yes, the voice is audible now.
Okay. Your slides are not visible. Slides are gone.
No, I have stopped sharing.
Okay. Okay.
I will share again.
Yeah. Uh,
Is it
Good
visible now?
afternoon to all. Uh, Good afternoon, all. Today is the seventh lecture in this Bheshaj Rachana Parishad, Drug Designing and Decoding in Ayurveda. For today's session, we have an eminent speaker and academician, Dr. Satyanarayan B. Sir. He Currently he is working as Principal Medical Superintendent and Director Research and Development at Munian Institute of Ayurveda Medical Sciences, Manipal. He has done his BAMS from SPM College of Ayurveda, Udupi, and MD in Bhaishajya Kalpana from IPGT and RA, Gujarat Ayurveda University, Jamnagar. He has worked as lecturer in the subject of Rasashastra and Bhaishajya Kalpana for both undergraduate and postgraduate students since last 21 years. He, he is working as also PG and PhD guide. He is also working as an R&D executive, MR for ISO in an Ayurved pharmacy, Munial Ayurved Research Center. Also actively involved in design and development of Ayurvedic formulations, quality control of Ayurvedic medicines, research on single herbs and herbo-mineral preparations, clinical research in Munial Ayurved Hospital under various Chikitsa Kalpa. He has published more than 20 research papers in national and international journals, also reviewer for 3 international journals. He has presented more than 75 papers in various national and international conferences besides chairing the scientific session. He has been conferred with Edward Kennedy Memorial Award to the best presenter from the senior research category, also bestowed with Shobhita Memorial Award. With this brief introduction, I request Sir to start his session. Sir, please start. Thank you.
Thank you. Thank you so much. Namaste everyone. Good afternoon. Shriyam Chadadyat Bhavatam Purarihi Yadanga Tejaha Prasare Bhavanim Virajate Nirmala Chandrikayam Mahaushadhiva Jwalita Himatram. Thanks for joining me. At the outset, let me thank the organizers, everyone from Shailya Rashashala and Shreyas Academy, and also more specifically Dr. Chinmay for inviting me and giving me an opportunity here to interact with all of you. The topic that was taken for today's deliberation is entitled as Design and Development of Herbal Formulations with related challenges and possible solutions. So let us enter into the topic without much delay. So when we talk about the term herbal medicine, globally there are various terminologies that are used. Maybe World Health Organization or many other international organizations are using certain terminologies. Before going into the subject specific, it is better for us to understand those couple of terminologies so that it becomes clear. However, I will be focusing on Ayurvedic products when I talk about design and development, but the term that we have taken is herbal medicines or herbal products. We have a term which all of you know very well, that is traditional medicine. It is the sum total of knowledge, skills, and practices based on the theories, beliefs, and experiences indigenous to different cultures that are used to maintain health as well as to prevent, diagnose, improve, or treat physical and mental illnesses. So it is the entire sum of the knowledge or skills or practices which may be based on certain existing theories, sometimes beliefs, and definitely on experiences. And they are indigenous to a particular culture or particular area, particular geography. And it is used to maintain health or for prevention purpose or for diagnosis purpose, to improve the general health or to treat certain physical or mental illnesses. In general terms, across the globe, traditional medicine is a comprehensive term used to refer both to traditional medical systems like say traditional Chinese medicine or our Ayurveda and Arabic Unani medicine, and also to various forms of indigenous medicine. The survey states that between 70 to 95% of citizens in a majority of developing countries, especially those lying in Asia, Africa, Latin America, and Middle East, use a traditional medicine. That may include traditional and herbal medicines for the management of health as primary health care to address their healthcare needs and concerns. It's also found out that even in industrialized nations, so-called developed countries, use of traditional medication is equally significant. In countries like Canada, France, Germany, and Italy, for instance, a report says that between 70 to 90% of their population have definitely used traditional medicines, maybe under different titles like complementary medicine, alternative medicine, non-conventional medicine, like that. World Health Organization has specifically defined herbal medicines. It states herbal medicines include herbs, herbal materials, herbal preparations, and finished herbal products that contain parts of plants or other plant materials as active ingredients. So, whenever we mention the term as herbal medicine, it can be only a herb or a part of the herb. Herbal material means extracted from a specific herbal part, or certain herbal preparations like, say, a powder or a kashaya, and finished herbal products like tablet or pills, ghrita or taila, that contain parts of plants or other plant materials as active ingredient. API or active pharmaceutical ingredient should be herb. Now the question is, why herbal medicines? Many people look for cures or treatments of certain unusual problems that modern medicine often does not address. Here, by the term unusual problem, I do not refer to those conditions which do not exist or people feels that he is having certain disease. It's not like that. These are the diseases which are having varied, mixed, complex pathophysiology, for which a modern medicine doesn't have a direct answer. If modern medicine does address these types of unusual problems, usually the solution will be invasive or very expensive. This is the reason why many people are going towards alternative paradigms, that is, natural remedies. Not only these herbal medicines or other conventional traditional medicines cure or treat such unusual conditions, it's important that they are also less invasive and more cost efficient. And very importantly, they have stood the test of time for their safety, efficacy, cultural acceptability, and lesser side effects. These products are widely accepted, maybe because of their cultural relation, or safety, which is well established because they are in use for several centuries. Their efficacy is observed in the field. However, we cannot say that it has no side effect. It's impossible for any product to have no side effect, but definitely lesser side effects than chemical medicines. Chemical constituents that are present in them. A herb has multiple chemical constituents, phytoconstituents. They are the part of physiological function of living flora. So it is believed that they have better compatibility with the human body than the synthetic ones. Ancient literatures have clearly
Doctor,
mentioned,
sorry to interrupt. Your slides are not moving.
yeah, that is right. I will share it to the whole screen, then hopefully it is possible. Just a minute.
Okay, okay, okay.
So I will share the entire screen. So you please check now. Is it moving now?
Yeah, yeah, yeah.
Okay, fine. Thank you so much. It's a well known fact that Ayurveda medicine is excellent in managing various geriatric conditions which are highly challenging today for entire medical community in the world. Ancient literature also mentions herbal medicines for age related diseases, maybe memory loss. It may start from early senile dementia up to the advanced Alzheimer's condition, or osteoporosis, diabetic wounds and ulcers, or autoimmune disease conditions, hepatic disorders, various metabolic disorders. So mainly non-communicable or lifestyle disorders for which there cannot be a better answer than herbal medicines, and especially the entire therapeutic protocol that is followed by traditional systems like Ayurveda, where modern medicine may be having only some palliative approach. So these drugs are made from renewable resources of raw materials. This is another important factor, which are eco-friendly. The process is also eco-friendly, and definitely it will bring economic prosperity to the growers. We can motivate growers to grow herbal drugs as raw materials, as input materials, which also helps in economic prosperity. It is well understood that India is one of the richest plant sources. We have at least 25,000 effective plant-based formulations, which are used in various traditional systems, starting from Ayurveda to Savarikpa, and also various folklore systems of practice, including the rural community practices. There are over 1.5 million practitioners of traditional systems in our country, and there are more than 8,000 licensed pharmaceutical companies. There can be almost equal number of unlicensed manufacturing units. More than 2,000 tons of herbs are being consumed annually. So this is a general picture which gives us a clear idea that how the herbal system is perceived across the globe and how important it is for us, and now the entire world is looking towards these herbal products. There were no existing guidelines for the design and development of herbal medicines. As per own idea, maybe based on some experiences, based on traditional experiences running through generations, based on certain scientific studies carried out in different centers, based on certain publications, the drugs were being combined and the proprietary preparations were being developed. But later, CCRS has published general guidelines for drug development, which is in 2 volumes. Probably you must have seen them. Otherwise, you can Google, you can get the entire dossier free of cost in PDF format. I'm not going for the details, you can go through that. A couple of points mentioned in initial few pages of this document are worth sharing. So that is what I am mentioning now. Here, most important thing that is expressed is quality control is a real challenge. They say that about 90% of products of Ayurveda are purely herbal. Of course, I may not accept this. At least, of course, maybe 70 to 75% of products that are used are purely herbal. So we have more than 10,000 Rasaushadhis, and I am not supporting those who call Rasaushadhis only as mineral or metallic products, because they are all herbal minerals. Herbs may be additional ingredients, or they may be used as a Shodhana Dravya, or maybe for Bhavana purpose and so on. So herbs are anyway exclusively used in all these kinds of formulations. There is no doubt about it. These plant drugs have different chemical constituents. A single herb has got hundreds of constituents, many alkaloids, tannins, glycosides, bioflavonoids. So many things are there. They may vary in same species of the plant due to influence of climatic conditions under which they grow, or due to the influence of nature and properties of the soil and fertilizer, geographical distribution, age of the plant, altitude, period of harvesting, storage conditions, etc. That is why we have all that beautiful scientific description for a collection of raw materials, where we define the references like Desha Anusara, Kala Anusara, Rutu Anusara, Vayu Anusara, etc. collection of the drug. This itself is a challenge for us, because now it is the industry. A physician is not collecting raw materials by himself and preparing. It is the industry now. Industry depends upon the drug suppliers who are supplying the drugs, who are never controlled properly. You will not be knowing from where they have collected, how they have collected, when they have collected. Whenever raw material reaches to you, you are writing that that is the supply date, and accordingly you are calculating the expiry. I will talk about this after a few minutes. So hence, to control the quality of raw drug material, raw plant material with reference to chemical constituents, if at all you are considering, is a tedious and difficult job. However, nowadays we are having metabolomic and also DNA fingerprinting technology that is applied very effectively, because chemoprofile is highly variable. A combination of metabolomics and chemoprofile or genetic fingerprinting and chemo fingerprinting is what we are following nowadays. Because of these reasons, plant drugs differ from that of conventional drugs, and some innovative methods are in practice nowadays for quality assessment. And it also includes composite study of Ayurveda and Siddha dosage forms. There are various dosage forms that you will have to consider, starting from Churna, Kwatha Churna or Sukshma Churna, Kashaya preparations, or Hima, Fanta, or Sneha preparations, Taila, Ghrita, or Avaleha, Paka, Kanda, Asava, Arishta, Sattva, Kshara, etc. types of preparations. Plant products and their derivatives, when we consider, in modern medicine also, there are 50% of modern drugs that are developed from plants now. So herbal drugs as a source for modern drug discovery has become very, very popular, even though we are not directly involved in them. There has been a quest to develop new drugs or formulations despite of being a costly and low success rate process. In recent years, the research, whatever is being done in drug discovery, it is more and more focused on herbal medicines or botanical source, by gaining lead from those traditional literatures, traditional practitioners, traditional experiences, and so on, because they have understood the importance of a reverse pharmacology approach. As traditional systems have very long history of usage of herbal drugs for the management of disease, definitely the success rate of their development as therapeutic approach is comparatively higher than synthetic counterpart, because we have a basic idea regarding their efficacy and safety. If you consider examples of modern medicines which are derived from the herbal source, that may be paclitaxel, Taxol, paclitaxel, they are derived from Taxus baccata like trees, used as chemotherapeutic agents to treat uterine and breast cancers. Vincristine and vinblastine, as you know very well, they are derived from Vinca rosea to treat leukemia. Artemisinin, derived from Artemisia annua, for which Nobel Prize was awarded to Chinese medicine a couple of years ago. Then camptothecin, which is derived from Camptotheca, a tree. Camptothecin is an effective anti-cancer drug. Podophyllitoxin, which is derived from Podophyllum hexandrum, another chemotherapeutic drug. These are all the drugs which are derived from the plant sources. There's a big story behind this, which is out of the context. I'm not going for the details. So, although drug development is driven by various such technological platforms, which are definitely helpful, it is a lengthy process in modern medicine. It requires huge investment. 10 to 15 years are required to synthesize a new compound. In 2006 itself, 16 years ago, the approximate average cost of a drug development was considered as 1 billion euros. And when this drug discovery and development process is done, half of the drugs are rejected at the stage of clinical trials. But sometimes soon after their approval also, phase 3 clinical trial is done, the drug is into the market, prescribed, the patients have started using them. Even in that stage, in phase 4 or post-marketing stage, many drugs were withdrawn from the market because of severe side effects and clinical risks that were not recorded during phase 3 clinical trial. If you remember the example of Rofecoxib, an analgesic anti-inflammatory drug that was launched in 1999, was withdrawn in 2004 because of a high risk associated with that to heart attack. The following modern approach of drug development is extremely costly, highly time consuming. That is why we have time tested traditional medicine, which gives you a reverse pharmacological approach, that is from clinic to the lab, not from lab to the clinic, and it is less time consuming also. So nowadays, new approaches to improve and accelerate the joint drug discovery and development process, and also to decide some drug target elucidation, lead target discovery, are based on a combination of modern methodologies and traditional information. Flash chromatography, high throughput screening, they have all revolutionized drug discovery. So traditional knowledge is a powerful engine here. Most of you have gone through this most popular article by Bhushan Patwardhanji, who has mentioned this particular picture in his articles. You can see the top, which is the traditional method of drug discovery in modern medicine with multiple bottlenecks, starting from a huge amount of molecules and ending at a very small number reaching to the drug market more than 10 years, and a reverse pharmacology where we have very good information you can narrow down to a safe and effective drug in 4 to 5 years. The drug development process typically follows certain aspects, which is again mentioned by CCRS dossier. And unfortunately, this description given in the CCRS guideline is also holding good for modern medicine. That is, allopathic drug or isolated active constituent from the herbs. It says you have to start with the isolation of bioactive ingredients and synthetic modifications of it, then evaluation of safety and efficacy, regulatory approval of the therapeutic agent if it is a new drug, then clinical trials. To carry out clinical trials, so you should get a regulatory approval. Then you have to carry out phased clinical trials. These are the components of drug development, again mentioned in the same dossier. You have to identify and prioritize the R&D needs, then go for the literature survey, then form hypothetical rationale based on the information in the texts, then initiate drug development, go for quality control, quality assurance protocols, then preclinical safety, check for biological activity and stability studies with the suitable standard protocols, then develop integrated protocols for clinical trials where good clinical practices, guidelines, and traditional methods are to be combined. You know that the GCP guidelines for Ayush drugs are also published. Then follow clinical trials with standard procedures, and due approval of regulatory authorities is to be taken. SOP for drug development is given here in this picture. I'm not going for the detail. It is very confusing. Again, whatever the details are given here are mainly for the isolated active principle that are applicable for modern drug rather than an Ayurvedic formulation. However, in general, when we talk about herbal drug development, it includes various steps that starts from a passport data on raw materials. Just like our passport, which has our photograph, name, full name, then address, age, sex, all these details. Similarly, for every herb, every raw material, there should be a passport data which speaks about the specific part, the time of collection, place of collection, its botanical name, local name, and so on. So it starts with that. A drug development starts with that. Then correct identification in relation to the passport data, pharmacognostic and chemical quality standardization, nowadays genetic fingerprinting, safety and preclinical pharmacology, clinical pharmacology, randomized control clinical trials. These are required even for Ayurvedic products that are proprietary Ayurvedic products. For them also, these steps are to be followed. When we go to the safety preclinical pharmacology stage, the guidelines may not be very stringent, but they are to be followed under Schedule Y. Now let us talk about the challenges that we will have to face in the field of herbal medicine. Problem starts at the conceptual level itself. As I already mentioned, herbal drug discovery usually follows a river pharmacological approach. We have to take a lead from the classical texts in Ayurveda, where you will find the documented information on several herbs and also extensive practical application and also the traditional wisdom. Even when you have to develop a proprietary product, each and every ingredient that is used in that product should have a reference in 54 authoritative books mentioned in Schedule 1. This is a well known fact for all of you. So, we should have a classical lead. We should combine the strengths of the knowledge base of traditional system like Ayurveda and dramatic power of combinatorial sciences, where several combinations and permutations of phytoconstituents can be made within minutes, and high throughput screening, which will help in the generation of structure activity libraries. Ayurvedic knowledge and experimental base with an experience database can provide us new functional leads so that time, money, and toxicity, which are considered as three main hurdles in drug development: time, money, toxicity. So they can be very well tackled. Problem starts at the level of raw materials itself, as I mentioned you in initial slides. Starts with the identification itself. There are lots of controversies involved. Next problem is with the substitutes. A few drugs are unknown, not identified properly. Many drugs are extinct. Many drugs are red listed, banned. Even the drugs like Sauseria lepa or Kushta or Katuki Picoriza Kuro, the trade is banned. It should not be exported. They are all CITES drugs, but we are using them extensively, and most of them are highly adulterated also. We have official substitutes. This is also something challenging. They have to be either having a textual base like Yoga Ratnakara, Vaishajya Ratnavali or Bhava Prakasha, and scientifically validated and approved. Collection is the major issue. You will have to consider time, place, maturity, official part, etc., which is not being followed. There are problems in drug supply authenticity, quality, and also the quantity. Your batch is about to start after 2 days. You need X drug in 1000 kgs, and you are supplied with only a part of it, a part is remaining. Or one particular drug, they assured you that they will supply, but they could not supply it. These are all the issues that we are facing regularly. Quality control is another issue. So, if I have to speak about the controversial drugs, most of you have definitely experienced this. A few examples I would like to quote: Daru Haritra, Berberis aristata, Berberis asiatica, Cusinium fenestratum are different plants that are used by the name Daru Haritra. Berberis asiatica is rarer, it is not commonly used. Berberis aristata is used, however, but it doesn't grow in all the places. It grows at high altitude. In Himachal Pradesh, it grows. In Uttarakhand, in high altitude area, it grows. So definitely, it is not available in the required quantity. And you know very well that its root, mula, is to be used. If you have to uproot the entire bush, then the plant will die. Cosinium fenestratum is the official substitute that is used in southern part of India, especially in Western Ghats, which is belonging to many spermaceae family. It is also endangered nowadays. So when Darvaridra word is used in the formula, you should confirm which particular drug is used. By the name Brahmi, Bacopa moniri is to be used. Hydrocotyle asiatica or Centella asiatica, that is Mandukaparni, is used in many of the places. For Pashana Veda, we have many plants that are selected, like Saxifraga ligulata, Erva lanata, Erva javanica, Amenia waxifera. We have Virginia ligulata, Bryophyllum, Coleus aromaticus, Rotula aquatica, Bridilla montana, Homoneia riparia, Ocimum basilicum. In our place here, it is the Erva lanata that is used for Pashana Veda. For Jivanti, we have Leptidania reticulata, and in some places, Desmotrichium fimbriatum is used, or Simicifuga fetida is used. Shankapushpi is the most controversial one. In southern part of India, in Kerala and a part of Karnataka, Clitoria ternacea, that is Aparajita, is used. In some places, Evolvolus alcinoides or Vishnukranta is used. In Bengal, like places, Canscora decussata is used. In Gujarat and other northern part of India, Convolvulus pluricalis, which is the actual official drug for Shankapushpi, is used. Rasna is another such drug. In Kerala, they use Alpinia galanga. In Karnataka, Vanda Roxburghi or Alpinia galanga is used. In northern part, it is the Pluchia lanceolata that is used. Even Viscum album, Withania coagulans, Ishwari, Aristolochia indica, Locnera, Enicostema, that is Mamejava, is also used for Rasna. Nagakeshara, which is becoming endangered nowadays. Mesua ferri has to be used. The pistils or stamens are to be used. Nowadays it is not available. The calyces are used many a times. It's substituted with Acrocarpus longifolius or Dillenia pentagyna. For Brahmadandi, Arjumon mexicana or Tricholepis glaberrima or Echinops. For Twak, this is another very important plant. Twak, we consider it as a so common, so popular drug. But in most of the places, for Twak, Cinnamomum cassia is used. Cinnamomum zelenica is not used. Sri Lanka is the best source for Cinnamomum zelenica, where the bark has a compound quill appearance. Even Cinnamomum tamala is used. Cinnamomum tamala is Tamalapatra, Tejapatra. Cinnamomum cassia is Patranga. Cinnamomum zelenica is Twak. This is a Barbaris aristata. Oxinium fenestratum as Daruharidra. Bacopa moniri. Hydrocotyle asiatica. Saxifraga ligulata for Bhashana Veda. Erva lanata and Erva javanica. Amenia baxifera. Bryophyllum. Rotula aquatica and Coleus aromaticus. Radiella montana. Ocimum basilicum. For Jivanti Leptidania reticulata, Desmostrichium. Simicifuga. For Shankapushpi convolvulus, Evolvulus alcinoides. Clitoria ternacea and Canscara decussata. For Rasa Vanda, Lucia, Alpinia galanga. Viscum, Anicostema littoral, Vithenia coagulans, Locnera rosea, Aristolochia. For Ragakeshara Mesophyri, Acrocarpus longifolius, Vilenia pentagyna. From Brahmadandi, Arjumon, Tricholepis, Echinops. For Twak, Cinnamomum tamala, this is Patra. This is the actual drug. Cinnamomum zelenicum, you can see typically this compound quill nature, where it is curved on both sides. This is Cinnamomum cassia patranga. So these are a few examples of the problems that we start experiencing at the level of identification and use of actual drugs. Then there is a debate on extracts versus crude drug usage. I have taken the excerpts from a publication by Dr. Naveen Chandra Joshi. You may be knowing that there are many pharmaceutical companies, I don't want to name any of them, who are running business in crores, are using extracts. Some of these pharmaceutical companies do not have a raw material storage section at all. They purchase standardized extract from the suppliers, and they mix them and they prepare tablets, or they mix them, percolate, and they prepare syrups. And even they do not carry out Sneha Pak also. They mix with the oil, agitate, and pack them. Extracts are widely used by many of the pharmaceutical companies, which are easy for them to standardize. This is a critical view regarding the use of extract versus crude drugs. Fortunately or unfortunately, use of extracts is accepted by Ayush licensing authority. We, however, may be using aqueous extract, water extract, or aqua alcoholic extract in the case of asafoetida, as the fermentation undergoes. From prakshepaka dravyas, alcohol soluble extract may come, or fat soluble extract in the case of Sneha preparations. However, Sneha or the liposomal drug delivery system, you will be having lipid and aqueous bilayer in that. Or in Kshirapaka, those materials which are soluble in water, soluble in fat, that can be entangled with the proteins, they form colloids and they can be extracted. Otherwise, we are not using any isolated principles. We strongly believe in using uni and polyherbal formulations where drugs are used as whole. However, in standardized extract, scientific validation becomes easier. It is said that these standardized extracts, say piperine 3% or curcumin 5%, vedolactone 3.6%, like that, it provides a consistent and effective alternative to crude drugs because they give the calculated ratio of active constituents. Concept of standardization now are widely welcomed across the globe, that is accepted as a rational way to accept medicinal herbs, even for physicians and patients. Even though drugs have got some active principles with which they work, it is extremely difficult for us to identify any single active principle. And nowadays, they have also understood this. Now, this is the era of network pharmacology. You may be knowing that. A particular phytoconstituent is not acting only on a particular system. It can act on multiple receptors, multiple biochemical pathways. So there are many such network pharmacological patterns developed with in silico studies in the case of Trifala-like drugs in treating various conditions like cancer. Now, however, the approach is changing, but there are certain active principles, there is no doubt about it, which are medicinally more important. There can be some other constituents which may help to counteract bad effect. One of the phytoconstituent may be useful as a bioenhancer. Another phytoconstituent may be useful to improve the shelf life of the product. Like that, every content for us in Ayurveda is important in a herb. So Ayurveda advocates that drugs should be used as a whole so that desired effects may be had without any side effect. We propound the principle of Rasa Panchaka, Rasaguna Veera Vipaka Prabhava, and we attribute the action of a drug to any one of these. Efforts have been made to preserve and fortify the potency of drugs, definitely in Ayurveda, by certain pharmaceutical processes and techniques. You may be compounding, you may be making the drug more and more finer, maybe grinding them, you may be generating the nanoparticles, you may be giving bhavana. The process of bhavana is an excellent process bringing about physical, chemical, biological changes to improve the bioavailability also, to improve the potency also. We are having many such pharmaceutical processes. For minerals, we have the process of preparing Bhasma, a very unique process by which you can improve the bioavailability, reduce the dose, improve the potency, reduce the toxicity, make the drug more acceptable to the body. So we have many such efforts. But evidently, Acharyas have refrained from isolating and using only the active principles. That is what we see. There is a well-known reference in Charaka Samhita Nidhanasthana, which most of you know. Prayogah shamayet vyadhim yo anyamanyam udirayet naso vishuddha shuddhastu shamayet yonakopayet. We always want to have a Shuddha Chikitsa. We believe in the treatment which gives a relief but causes no iatrogenic disease or no complications. For this purpose, it is essential for us to use the drug as whole, not isolated principle. There is another implication: treat the man as a whole and give the drug as a whole. This is mainly in relation with the Agni, which is responsible for the health and disease when it is vitiated. So the drugs selected should have its impact in improving Agni at different levels, maybe at Jatharagni, Dhatu or Bhutagni levels. Most of the Ayurvedic drugs, that is why, have Dipana and Pachana properties. If you see a formulation, you will find Maricha. They are all having Dipana Pachana properties. They are essentially needed to improve the bioavailability. In some of the formulations, you may wonder why these ingredients are mentioned. Those ingredients have their own impact. In Rasaushadhi, most of the Rasaushadhis are by nature are Depanapachana. A well-known statement is there: Swalpahi matra vipula gunascha sadyohi tad depanapachanascha. Sadhya depanapachana makes a Rasaushadhi quickly absorbable, assimilable and more bioavailable, giving you a quick action. If the active fraction alone is administered, we are not sure whether it acts on Agni, whether that is the purpose of using the drug as a whole. Third implication is that drugs possess not only the theoretical active ingredients, some other fractions which may be having their own impact to restore health of the individual. Some rasayana components may be there, some antioxidant principles may be there. When you isolate such principles and throw out rest of the drug, definitely we will be losing these beneficial effects. With all these concerns, we advocate to use drug as a whole rather than using them as isolated active principles. Now we have to face challenge in manufacturing processes. Variations are there in manufacturing processes of herbal products, maybe in drying or in storage conditions, which definitely affects quality and medicinal efficacy. Drying factors definitely influence. The temperature used definitely influences. It's difficult for us to follow sun drying method because of seasonal variation, possible contamination. When you follow modern method of drying methodology, microwave drying or freeze drying or vacuum drying, you will have to select which type of drying methodology is useful for which type of drugs. Storage also affects the quality of the product. Moisture reabsorption can happen, loss of volatile oil can happen, and if at all you are trying to inadvertently sterilize certain herbal products, that also can cause some effect. Herbs may be affected with molds, bacteria, insects during storage. However, follow GMP guidelines. Possibilities of adulterations may be there due to mishandling of the product or during harvesting, all these, which will have an impact on the final product. Once the composition of the formulation is finalized, then design the standard operative procedure and follow it. You will get the classical leads as in the case of Sharangadhar Samhita, where a step-by-step method of preparation is given for every category of formulation. For Avaleha, for Asavarishta, for Churna, for Kwatha, etc. You can follow the guidelines given in Ayurvedic Formulary of India, Part 1, Part 2 and Part 3. There is something which is described in a modern ISO approach, that is PDCA approach: plan, do, check and act. First we decide to develop a product. We do a market survey and we do get the information that X disease say diabetes mellitus or rheumatoid arthritis or ulcerative colitis or certain viral fever, doesn't have a most successful product, or the product available in the market are not so efficient, you have an opportunity to explore the market in this field. So when you get this information, a pharmaceutical company naturally plans and it decides that we want to prepare a particular product for this purpose. So we call the people of our company who are experts in that, the technical experts. We will ask them to get the formulation to develop. So they will refer various texts, they will refer the classical books, publications, scientific literatures, all this, and they will try to develop a formulation. So they will establish the objectives and they will decide the processes. These are the raw materials required. This is the place from where I can procure. These are the quality testing protocols that are needed. These are the manufacturing processes to be followed. This is the standard operative procedure. These are the equipments needed. This is the space required. These are the laboratory equipments needed to test the product. Everything you will have to plan, decide. So according to the objective, do the validation of process and equipments. You do the process, implement the process according to the plan. Then check whether you could do it effectively or not. Then act. If it is effectively done, the product is approved. If there is some problem, you will have to go back and repeat the process. Same thing is also mentioned as focus PDCA methodology. Find a process that needs improvement. For example, we developed a tablet X and we had all the plannings, raw materials, we have tested, we have processed them, we prepared the tablet. And once the tablets were prepared, they did not pass the friability test. It was getting powdered easily. Now we found the process that needs improvement. That is the process related to the hardness of the tablet and friability of the tablet. That means there is some issue related with the binding. So we have to organize a team which knows the process, who are experts in tableting. We will have to clarify the knowledge of the process. So by flow charting, this is the problem, this is the modification that we have to do. We have to change the blinding agent, you have to change the quantity, like that. Then do the process. Follow PDCA cycle again and see that whether it is improved. If improved, adopt it, adjust it, or abandon it. So this is how in a pharmaceutical company a product is designed and developed. Now let us observe whether we have any classical guidelines for this. In Charaka Samhita Kalpasthana, we have certain guidelines which you must have already studied. Faladinam pradhananam gunabhutaha suradaya. Faladinam here means Madana Faladinam, because this is the reference from Kalpasthana. Naturally, he speaks about those 355 formulations of Madana Phala. It is applicable for any product for that matter. Gunabhutaha means apradhanaha, suradayaha. In a formulation, you see that there is one major ingredient and many minor ingredients. It need not be there like that always. Say Rasnadikwata, Punarnavashtakakwata, where all ingredients may be equal. But Kutajarishta, Dantyarishta, Chavana Prashavaleha, Vasavaleha, Vasagrita. In all these formulations, we have one Pradhana Dravya. Tehi Tani Anuvartante Manujendra Vivetale. Just like a king is followed by his courtesans, action of a formulation is mainly decided by the main ingredients. Other ingredients follow the action of main ingredients. So when we decide the pharmacology or therapeutic effect of a formulation that we are going to design, to mainly concentrate on the major ingredient. Then it is said, Viruddha Viryamapyesham Pradhananam Abadhakam Adhikam Tulya Virye Kriya Samarthamisha. When you combine the ingredients in a formulation, they may be having Viruddha Virya or they may be having similar Virya. When you combine drugs are of Viruddha Virya, say in Kushmanda Avaleha we have Kushmanda which is Shita Virya, you may be having shunti like ingredients which are having Ushnavirya, but they are combined. You combine in such a way that action of main drug is not interfered. Adhikam tulya virya. If drugs of similar virya are combined, naturally the potency is increased. Say Chandrakala rasa, Bhavana dravya or Bhavana dravya or Shita Vira dravya, which makes it more effective in Daha. Then it is said, Ishtavarna rasasparsha gandhartham pratichamayam ato viruddha viryanam prayoga iti nishchaya. Why viruddha virya dravyas are to be combined? To attain Ishtavarna, to attain Ishtarasa, to attain Ishtasparsha, to attain Ishtagandha. Some prakshepaka dravyas are added, maybe to improve bioavailability, but to give good taste, good odor. You are adding sugar for one Ushtavirya dravya kashaya, maybe opposite virya, but when you bake it, you will get a semi-solid consistency, Ishtasparsha, that is required. So for this purpose, even though drugs are of Viruddha virya, they are utilized. Most important factor to be considered while developing herbal formulation is bhavana. भूयश्चैषां बलाधानं कार्यं स्वरसभावनैः सुभावितं हि अल्पमपि द्रव्यं स्यात् बहु कर्मकृत् तो स्वरसैः तुल्यवीर्यैर्वा तस्मात् द्रव्याणि भावयेत्. So when you give bhavana to a drug by using its own swarasa or its own kwatha or drugs with similar properties, smaller amount of the drug will give you more potent action, multitudinal action. As we give the example of Amalaki Rasayana, when Amalaki Churna is given the bhavana of Amalaki Swarasa, dose of Amalaki Churna can be reduced from 12 grams to 5 grams. Studies have been carried out on Amalaki Rasayana. Then bhavana given to Haritaki Churna with Haritaki Kashaya, Chaushashti Prahari Pippali. In all these, this factor is very well established. Bhavana is very useful in potensifying the product and reducing the dosage. There is another principle available for us. It is said, Vyadher ayuktam yadravyam ganoktam api tatyajet. Anuktam api yadyuktam yojayet tatra tadbudaha. We do find gana, like say Asanadi gana, Ushakadi gana, Varunadi gana, and so on. A gana has got multiple indications. The last few lines say that, say Varunadi gana, it is indicated in Ashmari, Antarvidrathi, Prameha, and so on. It is not that each and every ingredient mentioned in the gana is required for every indication. So Vyadhe ayuktam. If you feel that for the disease that you are going to administer, a particular ingredient is not required, you can remove that. Anuktam api yadyuktam, even though not mentioned, if you add another ingredient to the gana, if you feel that it becomes more useful therapeutically for the particular condition, you can do that. Say Asanadi Kashaya, which is popularly used in this part of country for the management of diabetes mellitus. Ekanayak or Celesia is used, which is not coming under Asanadigana. Anuktamapi yadyuktam yojayat tatra tadbuddha. This is a very clear liberty given by our acharyas that you can design and develop your own formulation. You can remove some components from the gana, you can add some components into that. It said, अल्पस्यापि महार्थत्वं प्रभूतस्य अल्पकर्मतां कुर्यात् संयोग विश्लेष काल संस्कार युक्तिभिः. You can increase the potency of a formulation by addition principle. You can reduce the potency by deletion principle. Say in the case of Vishadravyas, you may have to eliminate certain components so that it can be therapeutically administered. Say croton oil in croton tiglium. Or you may have to remove certain toxic components, say aconitin from Aconitum ferox, or strychnine, which may have to be converted into isostrychnine or brucin in the case of Vishamushti. So this is done by Vishlesha. So by Sanyoga or Vishlesha, Alpasya Maharthatvam or Prabhutasya Alpakarmatam is done. And this Sanyoga and Vishlesha are done Kala Samskara Yuktivihi, with adequate importance to the time factor, the processes that we do, and proper judicial planning. It said, Swabudhya iva sahasrani kotirvapi prakalpayat. According to the intelligence, thousands, crores of formulations can be designed because bahudravya vikalpatvat yoga sankhya vidyate. There is availability of thousands, lakhs of plants, several combinations and permutations. So you are at your liberty to design your own formulations. So these are very clear guidelines for us to develop our own formulations. Now let us talk about the problem that we encounter related to the standardization of formulation that we develop. So this is a process that manufacturers may use to ensure batch to batch consistency of the products. This is an essential measurement for ensuring the quality control. Standardization of herbal drugs is extremely difficult task. There may be some very preliminary parameters mentioned in the API or in pharmaceutical standards of Ayurvedic formulations or PLIM, which are not sufficient, and a few are erroneous also. I'm not going for the discussion related to that. There are many factors that influence bioefficacy and reproducible therapeutic effect. So in order to obtain quality oriented herbal products, we should take all the care right from identification of the plant with adequate importance to all the factors related to that: extraction, purification, rationalization of combination, all this. This is a dynamic process of revolution. For commercialization, correct identification and supply of raw materials to avoid adulteration has become a major challenge for us. There are many botanical species which are extinct. Lastly, the properties of botanicals recorded in the classics may have undergone changes over the period of time and environmental factors. And whatever the standardization protocol that are mentioned for modern medicine, that cannot be directly applied to Ayurvedic products. So that is why the problems started. All initial protocols, they were taken from the modern pharmacopoeia. However, now gradually we are incorporating our own standards. The concept of whatever active markers in the process of standardization that we are talking about should have a flexible approach, because these materials are highly complex in nature. The temperature, the grinding mechanism, environment, they can all change the active markers. There are certain thyla preparations where pippal is used, piperine was present in the raw material, final product did not show the piperine. How can you use piperine as a marker there? Anyway, piperine is okay. It is to some extent can bear temperature and stable. The components like tannogalic acid, it is highly thermolabile. You cannot use it as a standard marker for a number of Ayurvedic formulations wherever heat is involved. So our approach should change. Most of the reported side effects are there. They may be because of the extrinsic or intrinsic factors. Extrinsic factors like while collecting the drug, wrong identification, poor manufacturing process, contamination, substitution, all these factors. I'm not going for much details. When we talk about the herbal drug standardization, I already mentioned you that herbs are usually mixtures of many constituents. In most of the occasions, active principles are not known. Some markers may be known, but all these chemo markers need not be the active principles. So piperine is a marker and also active principle. Curcumin is a marker and also active principle. There may be many such markers which you may be using for HPTLC, HPLC or standardization, but may not have any therapeutic value. So in many occasions, active principles are not known. Then selected analytical methods or the reference compounds may not be available commercially. There are some markers, a few milligrams or a gram may cost 10,000 rupees or more, which is extremely difficult for a pharmaceutical company to procure, maintain it, and use it for all the batches. and many equipments, advanced equipments that we use for standardization. ICP-MS, AAS, HPTLC, HPLC, all these are definitely expensive and they are not commercially available for us. Plant materials are chemically and naturally variable, which depends upon various factors. Genetic variants are there, which lead to the variability of chemical composition. There are geographical and nutritional factors like altitude, soil composition, microbial load, climate, temperature. There are seasonal changes, rainfall, drought, seasonal changes and seasonal variation. In the case of Adatoda vasica, when the sample was collected from different part of the year from the same place and estimated, it was found that the plant which was collected in February March had maximum vesicin, whereas the one which was collected in August September had minimum vesicin. Similarly, the concentration of vesicin differed a lot from Manipal to Gujarat in various samples that is collected. So how you can standardize it is very, very difficult. I have many supporting articles, I will skip them. So this is a study on Desmodium gangeticum Shalaparni carried out by Jayanti and others. This study is about the variation in the phytochemical composition of Desmodium gangeticum depending upon season and region. HPTLC was followed. Lupiol was the marker component that was considered from the methanol extract of the root. The study has shown that percentage of lupiol was very low in young plants and it goes on increasing as the plant grows and it reaches to maximum percentage when flowering starts. In the case of geographical variation, it is seen that lupiol was highest in the roots collected from high altitude and lowest in the roots collected from the plains. In another example of Brahmi Bacopa moneri, where saponins or bacoposites are more important, highest saponin content was seen in rainy season. According to the age also, between 1 to 4 months, maximum saponin content was seen. In shoot, maximum amount was seen. So there are many such examples which are indicating us how difficult it is to standardize them. CCRS has given the protocol which are known to you. Protocol 1, 2, 3, 4, 5. Protocol 1 is regarding the standardization of raw materials with chemoprofile. Protocol 2 speaks about standard operative procedure of extract if at all you are using extract. Protocol 3 speaks about standardization of those extracts. Protocol 4 is what Vaishajya Kalpana speaks about, SOPO formulation. Protocol 5 is about standardization of formulation of finished product. It doesn't end there. Next is biological screening or targeted biological activity, including safety and toxicity studies, also preservation, packing, storage, including the stability data. So this is the protocol 1. So I will skip them. These are all available information and I'm sure that all of you know them. Test for heavy metals, microbial contamination, test for specific pathogen, pesticide residue, aflatoxins. This is protocol 2, protocol 3, protocol 4, 5 and so on. Now next, the challenges related to the monitoring of the safety of herbal medicines. If trade in a particular herbal product is made between different countries where regulatory status is changing, you will have to follow them. Herbal products categorized other than as medicines and foods are increasingly popular nowadays, like health supplements. Because when we think in terms of medicines, it should pass through very, very strict protocols. We have national regulatory information on herbal medicines. We have pharmacovigilance center where we can report the adverse effects that are observed. Physician, doctor or pharmacist, nurse, community health care worker can report. We can report at national or regional ADR centers. Suitable ADR form is available by National Pharmacovigilance Coordination Center at All India Institute of Ayurveda, New Delhi. This is the format through which you can submit the information. Now, what is the rationale for toxicological testing of Ayurvedic products? There are lots of talks and claims related with the toxicity of Ayurvedic products. Many published literatures are there. There are huge discussions, debates on the toxicity related with Guduchi recently. However, toxicity related with the metals and mineral products are being discussed for decades, and they were very well tackled also. So there are many issues, but whatever the studies are done at institutional level, they are minimal. They are not with globally accepted standards. It's very difficult to evaluate our polyherbal or herbo-mineral medicines using the conventional array of pharmacological or toxicological methods. We have to keep in our mind that there are many hundreds of such active ingredients. So a toxicity cannot be correlated with a particular ingredient. There are definitely many publications which state that phytoproducts have given potential toxicity. We cannot neglect them. Contamination by pesticides, herbicides, naturally occurring toxins, microbes, adulteration, synthetic substitutes, definitely they are the matters of concern. They must have caused adverse effects in many of the occasions. When you follow the standard operating method with all the hygienic conditions, possibility of toxicity is very less. So there is a need of a different fundamental approach of toxicity study in Ayurveda when compared to the modern approach. Toxicological protocols are prescribed by prominent agencies like US FDA, OECD, WHO. CCRS has given guidelines for herbal products and Rhasaushadhis. India also accepts OECD protocols. In our pharmaceutical company, we are following OECD protocol for acute, subacute, chronic toxicity studies. The type, nature, and extent of effect obtained during toxicity studies definitely will help us to adequately classify herbal medicines as non-toxic, moderately toxic, or severely toxic to the biological systems. There can be various categories, like category 1, where safety is established by use over long time, like classical products. Category 2, safe under some specific conditions of use. There should be well established document for this. Category 3 are herbal medicines of uncertain safety, where safety data is exclusively required, like new substance. As per the guidelines, ASU drugs, if they are classical products where ingredients are as per the classics, indications are as per the text, safety data is not required. Published literature is required, which is already there in the form of classical text. Pharmacological profile or clinical evaluation, schedule Y, is not required. If same classical formulations, same ingredients, but a different indication. Say for example, we have Rajapravartini Vati. If at all it is used for Shwitra, it is used by some practitioners. A new indication means safety data is not required because ingredients are same. Published literature is needed, but better you provide pharmacological and clinical data to support the claim. Now there are second categories, patent or proprietary drugs. If you are using the ingredients as per the classical text, there should be textual rationale. Safety data need not be given, but better to give acute toxicity data. Published literature will be ingredient based. You should have publication on individual ingredient. You need very basic pharmacological profile and very basic clinical evaluation of phase 2 clinical trial data. If there are any schedule E1 drug, Visha Dravyas, and a new indication, then safety data is required. Pharmacological profile, clinical evaluation, ingredient based publication is required. If any change is done in composition or dosage form or route of administration, one which is mentioned for external application, if at all you are using for internal administration, you need safety data, publication, pharmacological data, clinical evaluation, everything. Indian ethnomedicines, that is folklore drugs, which are not covered under this, if at all they are used, as per usage, they should have information. If available published data can be considered, you need exclusive safety study, pharmacological and clinical study. If at all extracts are used, aqueous extracts as per text ingredients, new indication. In all these, safety study may not be required for aqueous extract, but pharmacological study is required, clinical study is required. For hydroethanolic study, you require safety study, pharmacological study also. Even if it is any other extract apart from water and hydroalcoholic, that is also accepted nowadays. But you should carry out extensive safety study up to chronic toxicity study, even teratogenic study also, and pharmacological phase clinical trials. This is what the guideline states. So with this, let me conclude. Sorry if at all I have taken more time. The material was unequally distributed as I didn't have time to format them properly. So there is no doubt that herbal products have their own advantages. This is the era of globalization. We have to popularize them across the globe. We have to understand the global guidance. With a commercial point of view, there are many challenges that we have to face. And the major challenges are related with the variability, lack of standardization, poor documentation of safety and efficacy studies. To overcome these challenges, we need to adhere to BACP, good agricultural and collection practices, GMP, good manufacturing practices, TLP, good laboratory practice standards, and definitely good clinical study practices also. Establish appropriate specifications for raw materials, intermediates, and finished products. Comprehensive documentation on manufacture and testing. You should document every point in your manufacturing control sheet. The starting date, the drug taken, part taken, the washing time, who was in charge, how it was washed, drying temperature, how the powdering was done. Everything should be documented very well. Safety and efficacy studies as per standard protocol. These are a few of the references that I had considered while designing this particular presentation. So thank you very much for your patient listening. Thank you for the opportunity.
Thank you very much, sir, for this elaborative session. Challenges in front of manufacturing sector, herbal medicines, and how to overcome it on it. Thank you very much. If anybody is having question, you can ask in chat box.
One question I can see, I think here. Every herb has its own dhatu gamitva. What is the principle behind that? This is nothing but the concept of targeted drug delivery, which is mentioned in modern pharmacology. When you study pharmacodynamics and kinetics, you will understand the receptor specific theories. Very interestingly, very beautifully, this is also applicable to our herbs. I'm not talking about any isolated phytoconstituent. An individual drug also has got an affinity to act on particular dhatu, particular dosha, and also a particular srotas. So this is a specific affinity related with the receptors. It may be receptor agonist, it may be receptor antagonist, If it is agonist, it may be having different type of interaction, maybe a mechanical interaction or a chemical interaction. The entire receptor theory is something that we will have to address here. And the second thing is related to the nature of the drug molecule. Depends upon its physico-chemical nature. Mainly, it is based on the DNA parameters with which we can evaluate the drug also. There are DNA fingerprinting technique where ISSR techniques, scar marker techniques are used to identify those markers. Those markers are also having a specific gene tagging ability. They can tag with the particular protein carriers and show the action. So every herb definitely has got a concept of Dhatugamitva. Of course, in relation with Ayurvedic principles, you can describe based on the principles of rasa, etc. Even with that, also, I would like to mention you one of important research works that have been carried out on extraoral taste receptors. Some of you must have read those articles. That was an Indo-Romanian collaborative work. Concept of rasa and vipaka have been very well established there, even including the concept of virya. So here in Ayurveda we see that these are the madhura dravya, these are the tikta dravya. Gana we are having according to rasa also. We are saying that madhura gana dravyas they are having brahmana, rasayana, balya like properties. It may have sharkara in it may have shatavari in it may have vidari in it. When you analyze it, chemically or phytochemically, it may be having totally different constituents. A modern pharmacologist will laugh at us that how they can have similar actions. But very interestingly, studies have shown that there are some taste receptors which are present outside the tongue also. They are called as extraoral taste receptors. In stomach, we do find madhura or sweet taste receptors. In liver, we do find bitter taste receptors. We know that Tikta Rasa Dravyas act on Yakriti. Madhura Rasa Dravyas, they can act on Amashaya, Kafasthana. There are many such observations which have given a very clear indication that probably gustatory taste receptors are related to rasa, extra gustatory are related to vipaka. This also gives you some idea related with the dhatu gamitva or dhatu specific action of our products. So our Rasapanchaka concept is highly advanced. As the modern investigations are getting advanced, we are getting more and more information related to this specific targeted actions. There is a question why Raja Pravartini Vati is used in Shwitra. However, that is not my clinical practice. Uh, it is used both externally and internally in Shwitra. There is a practice locally by some of the practitioners where they grind Raja Pravartini in lemon juice and apply. There is a reason classically also to this. When you observe the ingredients, there is an important ingredient here, that is Kasisa. Kasisa has got a specific indication in Shwitra. If you read the literature from Rasa Shastra, Kasisa is a Shwitagna Dravya. Both internally and externally, Kasisa can be used in Shwitra conditions. But in local practice, they are using it externally. However, you can try it for internal administration also.
If anybody is having still any question, else we can end up with this session. Thank you very much, sir.
Thank you very much.
Uh,
Thanks for joining me. See you again.